Hello, everybody, and welcome back again. Welcome to our first FRQ review. So today is going to be a little bit different than how things have been in the past. Today we are going to start looking at exactly what we were going to do on the exam. So instead of just looking at more at content specifically, we're going to be looking at more at the exam specifically. So a little bit different. As people are coming in today, we're going to go ahead and do a poll to get us started. My voice is feeling a little bit raspy today, so I do apologize for that, or if I am coughing at all. So I do have my favorite baseball team cup here with me today, Oakland A's fan, if anyone else out there is an Oakland A's fan. Or let me open the chat here. If you are a baseball fan, throw your team name in the chat. Okay, and as we start, so today we're going to be talking about the FRQ that goes over like scientific investigations, which is basically being a scientist in general, and the process that follows and what you would need to know, in order to do investigations. So that's what we're going to learn about today. So I thought it would be fun to throw this poll here as people are coming in. What type of scientist would you be if you could choose of these four options? Would you be a marine biologist, an astronaut, a paleontologist, or an infectious disease doctor studying the spread of infectious diseases? So go ahead and choose one there as we are coming in. I'm going to go ahead and share my slides with you. Up here, make sure this fits. Take another second to answer. Make sure this takes up the whole window here. Gets to like a certain size and then it gets smaller. Okay. Let's publish our results here. Oh, we are unanimous. Man, you guys are right after my own heart there. We are 100% unanimous here for marine biologists. And I don't know if I told you guys this when we first started. I know I talked a little bit about my background, but my degree is actually in marine biology, and I went down that route for a while to be a marine biologist. I actually worked at a few different aquariums doing the whole dolphin show thing, and then definitely wanted to get out of that realm of things. I don't know if you have kind of seen that there's been more of a movement as far as moving away from keeping cetaceans in captivity, and I'm definitely, after working with them, a firm believer in that, and so moved more into the education world of it. Worked at the Monterey Bay Aquarium for a while, and then went back into teaching too. So I am excited that I see so many marine biologists out there. You can definitely do it. If that is what you want to do, pursue that for sure. All right, let's get into it. Sorry. So again, if you don't want your name to appear in the chat, and we can all see your name as you're talking, you can just use a different name or a pseudonym if you don't want us to see what your real name is. And here is a video. Let me make sure the sound is up on this. This is a video. This is our Fivables co-founder, Amanda, and she's going to talk to you about a new social media challenge that's coming up. So let me make sure the sound is working on this here.
What up, AP students? It's Amanda here from Fivables. We are literally giving away $100 to pay for your AP exams to like 50 different students. How do you win? Easy. You just got to post about the AP experience on social media, on TikTok, Instagram, or Twitter. Use the hashtags YesWeCram and with Fivables, and tag at Fivables. So one person will win every single day, and this is how you can win. It is going to be a group of students who actually judge the winner. They will pick the winners, and this is how they're going to choose. It has to capture the AP experience. Do they look at that post and think like, that's 100% what this feels like? Is it creative? Is it funny? Does it have a really awesome song in it or really good use of some sort of meme? Anything? Do we see creativity and engagement? How many likes do you have? That literally is going to help you win. The more likes, the more likely you are to win. And then finally, does it incorporate Fiveable? Is it in the caption, what you're wearing, in something that you're doing? How have you incorporated Fiveable into it? So this is how you can win. A couple of quick notes. Make sure you follow Think Fiveable so we can DM you if you've won. You can post as many times as you want. You can post today, tomorrow, every day, as many times during the day as you want, but you can only win once, so keep that in mind. And finally, some of our best social interns came from this contest last year. So if you're really awesome at social media and you're looking for an internship, show us what you've got because we want to hire you. So I can't wait to see your posts. See you on the flip side. Good luck on your exams. Talk to you later.
Awesome. Yeah, definitely. That's what I've been talking to you guys about too. So if this is your jam, if you are,
what up, AP students? Amanda here from AP students?
Oh man,
Amanda here from Fiveable. We are literally giving away.
There we go. Let's move on. If you are into social media and into posting and creative, and if that's your jam, definitely do this. We are always hiring students to come on and help us in different aspects, whether that's to be TAs or to be part of our social media team, then definitely do it. Let us know you're interested and you want to be part of the team. We'd love to have you. Okay, so we have just 2 more hour-long crams after this, and then we have our finale together, and that is on the 13th. I will be with you for 5 hours that day. And we are going to go over, we're not going to go over all the content again because we just did that and you can rewatch those videos whenever you want. So we are going to do it more like we're going to go over FRQs and then we're going to review the content that was in them. So we are going to get some content, but not all of it, more targeted test prep for that. So definitely make sure you've got your content kind of down before we do that. Not to say we will go through some content, but we're going to try and like play some games and do some multiple choice and that kind of stuff a little bit more so in that finale. Okay, so today we're going to do some general FRQ facts. We're going to go over type 1 and we're going to do it a little bit differently. The AP exam switched kind of what it looks like, the environmental science exam switched what it looked like in 2020. And then of course, 2020 just like exploded and we weren't able to really do the exam anyways. So this new type of FRQ, it's hard to find a ton of examples online for you to go over. And so I think what's going to be better, instead of trying to like do exactly what you're going to see on the test, I think we should just do a more solid foundation of what is an experiment and how to build an experiment and really know what that is. And then that way, when you have a question asking you to analyze one, you'll feel comfortable in that. So I think for today, that is, the strategy we're going to take. So it is a little bit different. We're not going to look at something that would look maybe exactly like what you saw on the test. But what I'm hoping is that I'm going to give you so much knowledge on experimental design that when you do see that question on the test, you're going to be like, oh, this is easy. I've got this. So that's my strategy, because I think that there's not enough really examples of what you'd see on the test that if we tried to do it, we're going to miss the mark and then we would have kind of like wasted our time. So I think that this is a better strategy. So again, section 2 of the test is the free response. There's going to be 3 questions. You have an hour and 10 minutes. It's 40% of your total exam score. Today, we're going to go over question 1. And then the next 2 crams we have, we're going to focus on question 2 and 3. And those ones we do have more examples for. So we will look more at exactly what it's going to look like on the test for those ones. But today, question 1, design an investigation, presents students with an authentic environmental scenario accompanied by either a model or a visual representation or quantitative data. So what in the world does that mean, right? Let's break that down. First of all, and we're going to go over this like in the next couple of weeks too, just to make sure you've got it down, 70 total minutes. I mean, you can have about 23 minutes for each question. Time yourself when you practice. I think that that's key. You can spend more time on one question than another. So you don't have to spend exactly 23 minutes on each question. You can spend, half an hour on one question if you want, and then 10 minutes on the next, if that is easier for you, whatever works best. And you can complete them in whatever order you would like. Oh, also, shout out to Miriam, guys. Miriam is our Environmental Science TA, I am sure you are familiar with her. Say hi to her in the chat and feel free to ask her any questions you have as we go. All right, the new thing too with the test this year, again, this was like new in 2020, but we kind of didn't get a chance to really use it, but you can have a calculator for the entire test, which is awesome. So you won't have to be stressed about doing like simple math or things like that that are going to slow you down. We'll be able to speed right through that. So we can focus more on, what equations do we need to learn for this class, which there's not really a ton, but there are some that we should make sure that we're ready for. So we will have that. That's not really on this FRQ question, but we'll make sure we know that moving forward. All right, and so this one, I don't want to spend too much time on this, and you can see this, it's kind of in like all the AP classes. It tells you exactly what each of these words mean as far as what they're asking you to do on the exam. So like when they want you to calculate, what does that mean? When they want you to describe something, what does that mean? So I really, this slide is going to be in here for you guys to reference again and again. Again, I'm going to put it at the start of all of our FRQ crams for you, but I would definitely take the time to read all of these examples and think about them and make sure that you're ready for when you see on the exam, it says, make a claim about something, what they want you to do there, right? So definitely take the time to look at these more kind of past what we are going to do here today and make sure that you feel really comfortable with these words and what they're going to be asking. All right, so we are going to jump right in it, talking about scientific method and what does that mean and what are the key words that we need to know for that. And that experiment, well, it's not really an experiment. It's a good example of something that's not an experiment. It's called elephant toothpaste that they were doing and exploding there. Let's maybe go back to that for a second, talk about that. So a lot of times when, we're thinking about class, and especially if you think back to like your younger years when you were maybe in like elementary school and doing an experiment, a lot of times it wasn't an experiment. And that's important for today and what we're going to look at because of how you need to design something for it to count as an experiment. A lot of times when we do something like this would be a demonstration, right? Because it doesn't have a hypothesis. It doesn't have two groups that you're comparing to each other. It doesn't have like different variables going on. This is just a demonstration. So a lot of times, especially in like younger classes of science, we'll do something, call it an experiment, but it's really not. So let me give you another example. You may have, like when you were younger, done that experiment where you put like celery in different colors of water, and then you see like the water, the color, the food coloring come up the leaves of the celery. And that's not really an experiment because you're not really testing anything. I guess it could be if you were saying like, your hypothesis was that red food coloring will travel more efficiently through the celery than blue, and then you'd have to have some sort of way to significantly measure that. But if you see where I'm going here, in general, a lot of the things that we did when we were younger and we called experiments, they weren't. They were just like a demonstration. So what makes it an experiment? It has to have different variables. And that's where we start looking at these. And again, it's always difficult when you have words that kind of like look the same to really pull it apart and know how those things are different. So independent variable and dependent variable, you absolutely have to know how to identify those in an experiment, right? So independent is changed by the scientist and dependent is what you measure. Again, the independent variable is going to be changed by the scientist. So it's going to stay, in different increments or things like that. And then the dependent is going to, again, it depends on the independent. So it is going to change. And that is what you're looking at and then measuring. So in our celery, if that really was an experiment, you'd say your independent variables were your different food colorings. And your dependent variable would have been your degree at which they went through the celery and appeared at the top. And so you'd have to have a way to adequately measure those things. And then your control is a variable that's not changed. And the control happens kind of in two different places. You're going to have a control version of the experiment. So let's keep on a roll here with our celery example. You would have a piece of celery in just plain water, right? Because if your piece of celery that was in plain water, if the leaves of that celery turned purple, you would know that something weird happens when you just put celery in water, and it actually has nothing to do with the food coloring, right? So that would be a control variable. But there are other variables you'd want to control as well. Like you wouldn't want your blue food coloring water to be boiling and your red food coloring water to be icy, because that might cause, different effects. And again, that's an extreme example, but still you'd want to control the temperature. You'd want it to be all the same. You'd want them to be in the same amount of sunlight. You'd want the celery sticks to be of about the same size. You'd want the amount of time that you left them in the water to be the same too. So those are all different variables that are controlled. And it's important to control every single thing else other than the one thing that you want to test. Otherwise, you're going to have your experiment, you're going to not be sure what caused your dependent variable to change. So you're going to have to make sure that you control every other variable going on in the world around your experiment to make sure that it's a good experiment and it really measures what you want it to measure. All right, so the next thing then is your hypothesis. So a hypothesis is not like an educated guess. I know that that's kind of what we were taught when we were younger, but that's not really right. A hypothesis is going to be a prediction or a statement about the results. So that would be after you've done a lot of research. You would make a statement that says, well, I know that red food coloring particulates are smaller than blue. So my hypothesis is going to be red food coloring will travel more efficiently to the leaves of the celery, right? Whereas then your null hypothesis would be anything else would happen. So your null hypothesis would incorporate basically not that happening. So your null hypothesis would say the red is not going to travel to the leaves faster than the other colors or more efficiently than the other colors. Then your null hypothesis would then encompass the idea that maybe the blue would travel faster, or maybe neither of them would travel at all, or they would travel at the same time, or the plain water would turn it purple. So null hypothesis is really going to capture any other possible outcome other than the one you think is going to happen. And then at the end of your experiment, you either accept your hypothesis, like it did work, it was the red, or nope, it didn't, it wasn't red, it was something else that happened. And then finally, qualitative and quantitative data. Man, talk about two words that look the same, and it's hard to pull out here. So quantitative is from the word like quantity, and that's going to be numbers. It's going to be something that's graphable, right? Qualitative, you can think of quality time. It's more of like an emotional feeling, maybe. Like maybe you could say that one did it a lot and that one did it a little bit, right? So it's not really measurement, but it is, it's more of like an idea, like that, made me very scared and that one made me not scared, or, it did happen or it didn't happen. Not things that you can really graph, right? And sometimes maybe not as accurate or not as scientific. So quantitative data is going to be data with numbers that can be graphable. And if you remember, think back to just now when we were talking about this FRQ, it said that it was going to have quantitative data, right? So we're talking about an experiment here that's going to give you some numbers, which I imagine they would either want you to interpret a graph or chart, or you're going to have to make a graph or chart. So let's keep moving right along here. Oh, right. So this is my favorite way to teach the scientific theory, the scientific method. So we are going to watch a quick snip here of Mythbusters. And out of this, we are going to then pull the hypothesis, null hypothesis, independent variable, dependent variable, controls, what type of data are they gathering, and potentials for scientific error. So as we're doing this in the chat, if you could start to pull out some of these different things as you see them. So you could say like hypothesis is, and then like write what you think their hypothesis is. You could say, oh, independent variable, and then write that down as we're going. So as you see these things start to come up in this video, throw them in the chat if you feel comfortable.
With Pliny the Elder's reputation on the line, it's all systems go.
We've chosen this area because it's open enough that we can see what's going on. And we know there's some food over in that direction. And we're hoping that the elephants will come through here on their way to the food. And at the right moment, we'll roll over the ball of dung, release the mouse, and see what happens. Hey, Adam, are you done yet?
I'm all set, ready to go.
All that's left are some fairly predictable predictions.
For me, I see this the same way I would see a person looking at a fly on the ground. It's going to be so small that you may not even notice it. I don't see any reason why an elephant would be afraid of a mouse.
Honestly, I don't think much is going to happen. I don't have high hopes for this myth.
With sensible money on the no reaction outcome, it's ready, steady, go. The elephants innocently approach the dung of doom, and Adam and Jamie prepare for the seemingly inevitable outcome. But then the elephant backs away, leaving the Mythbusters flabbergasted.
What just happened is a classic Mythbuster scenario. We were all like, yeah, right, this little guy's going to scare those big guys. And it did. I mean, our jaws just dropped because it was clear as day. The mouse came out, the elephant stopped in its tracks, like, whoa, and then it gently tiptoed away as if this thing was deadly. We all thought this was never going to happen, yet there it was.
All right. Okay, so let's go down this now. I see some people dropping some stuff in the chat already, and that is awesome. So let's look at our hypothesis. What was their hypothesis? And it was that the mouse is going to have no effect on elephant behavior, right? So they said their hypothesis is that the mouse will have no effect on elephant behavior. You could say because it is of such an insignificant size that the elephant won't even notice it. And then their null hypothesis would have been anything else, right? So like the elephant gets scared, the elephant like could eat the mouse, the elephant could, you know, any other thing happening than the elephant not noticing, right? So the null hypothesis is what they would accept. They'd say that they accepted their null hypothesis and that the elephant its behavior was significantly altered by the mouse, right? I don't know if it was scared, maybe. It kind of looks scared, looks startled for sure. So yeah, definitely going to accept their null hypothesis on this experiment. What was the independent and dependent variables on this? Independent and dependent variables. And again, if you're watching this in a replay, definitely be thinking these, saying them out loud, thinking them in your head. What do you think these were? Remember, the independent is controlled by the experimenter. So the independent variable here would be the mouse, right? So they showed the mouse, and that the dependent variable would have been the elephant's behavior, right? Is this, we can jump down to type of data. Is this quantitative or qualitative data? This is definitely going to be qualitative data. Good job, guys. It's not something that you could like graph, right? Unless you were somehow putting a number on the elephant's reaction. But really, this is just, did it alter its course where it was walking? And it definitely did on this one. So that is going to be qualitative data. Controls for this one. Now, let's kind of talk about controls and potential for scientific error kind of at the same time here. So they would have had to, and I think if you watch the full episode, they do it more than once. So like, what if just that elephant is afraid of mice, right? They need to do this experiment with multiple elephants to make sure that they're actually afraid of mice, right? So you're going to have more, a bigger sample size for sure. So, and a control would be, let's have another elephant walk through here to get to the food, and let's pull the little dung ball over with no mouse in it, right? Because what if the elephant is just afraid of the dung ball rolling over and it has nothing to do with the mouse at all? So that's their potential for error and a control. So they would have to do the experiment again, either with the same elephant or with different elephants, and then pull the dung ball over and see if they're just afraid of that. Any other potentials for error that you could see here? I definitely saw like the string. I know it's like a fishing line they were using, but that could freak it out. The dung ball rolling in general, I think, would freak it out. They used a white mouse. Maybe elephants are just afraid of the color white. What if they do the same experiment and they just put a white piece of paper under the dung ball, and it's afraid of that, right? So if you look at this experiment kind of like at the surface level, it seemed like it was really a good experiment. But if you look at it, like break it down a little bit more, there's a lot of potential for error. And so a lot of things they'd have to do in order to prove their theory, right? And prove that it was a good experiment. Ooh, good note too, Miriam. Maybe they're too close. Like they're standing right next to these elephants. And these were elephants, if you watch the full episode, they're like from a reserve. So they're used to humans. And so we're assuming, I guess, in theory that they are not afraid of humans, but they're wearing like weird sunglasses and hats and stuff. Like maybe they're just afraid of the Mythbuster guys, right? So we definitely would want to look at that too and minimize the human impact being there as well. Awesome. Okay, I want to make sure was not too much in the streams. Let's make sure we have enough time. We're actually going to do one more. How does it always do that? Let's go like this. There we go. Okay, we're going to do one more. So as we go this time, just start throwing stuff in the chat right away when you see it in the video.
The age-old scientific myth that spider silk is stronger than steel is coming down to the wire. In their initial small-scale test, 500 strands of spider silk proved to be twice as strong as its steel equivalent. And now the Mythbusters are ready to take this test to an unprecedented scale.
There we go.
To test the strength of a 28-gauge steel wire to its matching equivalent in density, a whopping 25,000 strands of spider silk collected from 42 spiders over the course of 5 weeks.
This is quite possibly the largest amount of spider silk that's ever been brought to bear on a test like the one we're about to do, to compare this to the strength of steel of the same mass. Why go to this length? It's a great question. The fact is when you're testing stuff in scale, as long as you iterate, you can come to some good conclusions, but the best way to get great results is to experiment in the largest scale you can, hence this monster.
And for this final silk versus steel smackdown, the team has adapted the test rig with a clever addition.
With our new rig here, we'll be conducting 2 different experiments. The first with our length of steel twine here, with our water being released in a controlled manner down to this bottom tank here, and it will weigh down that cable until hopefully it'll sever and we'll be able to see how much weight in water is causing that thing to snap. If the results of the previous test hold true, I'm pretty sure that spider silk is going to kick steel's butt.
Cue the butt-kicking attempt. The 28-gauge wire's up first.
Steel wire test in 3, 2, 1.
They've traded out washers and paper clips for over 40 pounds of water.
It's so amazing.
So quiet.
Gravity does its job, and as the water trickles into the lower bucket, the steel wire starts to suffer from some serious water weight gain.
It's
Oh,
stretching.
wow, look at that. It's really moving.
It's stretching.
It's visibly moving. That is crazy.
I heard a noise.
Oh, oh. That was fantastic. That couldn't be a better result. All right, let's weigh this puppy. 12.5 pounds, Cannon. That's a result. Let's set up for spider silk.
Definitely.
25,000 strands to be exact.
There we go.
It's a nice yellow color.
I'm gonna let it take on the weight. This is the most amount of spider silk that's been tested in this kind of test, maybe ever. 9.5 miles of silk right there.
That's crazy.
9.5 miles. That's insane.
You guys ready?
Totally.
Yep. 25,000 strands of spider silk in 3, 2, 1.
Releasing the water.
Cord by cord, it's now stretched about a full inch.
The 25,000 strands of silk bears the load.
Okay, we've passed the weight of the steel.
Whoa.
So,
The silk has surpassed the steel's holding power,
We're coming up on double.
but the strain of the weight is taking its toll.
What was that?
that was
Inside snapped.
Whoa! Ah! That's a result!
Oh man, that was cool.
That was fantastic. All right, our steel wire held 12.5 pounds. This is the amount of water it's equivalent in mass in spider silk held, and it is
Cue the drum roll.
26.
26 pounds! That is so cool. Aren't you excited?
Yep.
Here, give me a high five. We are so psyched. Can you tell?
Just as it did in the previous density equivalency test, the spider silk proved to be a little more than twice as strong as the steel. 2 head-to-head density tests, 2 almost identical results. A satisfying conclusion to this classic scientific myth.
This is definitely one of my favorite stories this season. No fire, no bombast, just 9.5 miles of spider silk and a set of really elegant methodologies for testing its strength. And you can picture in your head what a fine steel wire feels like jeweler's wire. You can feel that. Okay, now I want you to hold that in your head and watch this. This is so gossamer, it is barely even there. And the idea that it held twice as much weight as the steel wire, mind-blowing. All right, Mythbusters.
All right, I'm going to stop that there for us. So you guys have thrown some awesome stuff into the chat so far. You're really hitting all of this really well. I hope you're enjoying this idea of going over experiments. It's a really cool way to kind of test your thinking of it and being able to identify it. So the hypothesis in this one was that spider silk of the same mass would be able to hold more weight than steel. So that's their hypothesis, and their null hypothesis would be the opposite of that it wouldn't. Really it would be that the steel would hold more for this one, because we're just comparing two things in this. Our independent variables are the cords, right? So one is spider silk and one is steel. So the independent variables would be the cords or the attachment pieces. And then the dependent variable would be the total weight or amount of water that it was able to hold. In the control group of this, so yeah, because we're really just comparing two separate things. Our controls that we're really looking for are things in the experiment that were done to maintain consistency. So that would have been the fact that they used the exact same bucket, they used the same system to put in water. They would need to have put the water in at the same rate, because if you dumped a ton of water in one and then did the other one slowly, that would create a different velocity. So again, the water would have to go in at the same rate. I don't know. I think someone else said some more stuff, same bucket, same water. Yeah, all that. So type of data, this one is going to be quantitative. Again, they had a solid number. You could definitely graph that for them. If you were going to graph this, it would probably be like a bar graph. And you would have your two steel and spider silk on the bottom, and then you would have the total weight on going up towards the top that you'd be able to graph. Anyone see any potential for scientific error? This one's a pretty solid experiment. And it was the same mass of the two, the steel or the spiders. Let's see. The only thing that I could really see as a potential for error in this one was like that little attachment piece that they had on the spider silk. Like, to me, was that making it stronger or less strong? And like also the fact that the spider silk was only like, I don't know, maybe 8 inches long, and then the wire was like really long. So like, wouldn't it have been fair then to like wrap the wire around a million times in a row? Right? Like, it didn't seem fair that you got to wrap the spider silk around a bunch of times, so it was like really thick next to each other, whereas the steel was just like one little tiny string. To me, that seems like not right. So I don't know. Think about that maybe as far as like, was that a good experiment? I don't know. Not sure. Okay, so cool. Awesome to think about. Really good way to kind of use these ideas, because that is what I believe is going to come up on your exam. They're going to want to know, or have you break down, oops, why does that always start over? Have you break down an experiment to these different parts. So I'm going to have you do one now, not from a video, but from reading it and breaking this down. So again, there's 6 questions here. If you have a piece of paper near you, grab something real quick and write down these answers as we go. I'm going to give you 10 minutes on the clock really quick here, and then we will see what you got for your answers. And you might be thinking like, man, that's not going to take me 10 minutes. But that last one there, number 6, is I want you to graph the results of the experiment very quickly, rough graph, okay? It's fast, kind of throw this together in our 10 minutes here. It doesn't have to be beautiful or anything like that, but I want you to experiment, or I guess practice with reading and pulling the information out. All right, guys, just take these last few seconds here to finish that up. Ooh, it's not like that alarm going off. Okay, so we are all done here. What I'm going to do, let's see if I can pop this out of presenter mode for us here. Here we go. And I have our answer slides. I'm going to enter in right here. That way they will be here for you if you come back and check this out later. So let's go back in the presenter mode. And let's get our captions going. Get our captions going again. Okay. Here. No. Stop it. Here are our answers. Okay. Definitely, like, whenever we do this, we're going to continue to do this for the other FRQs and then in the finale too. So make sure you have paper, ready to answer these. And actually, for the next one, I thought it would be really cool if you guys wanted to answer it digitally. And what we can try and do is have you upload your answers anonymously into a Google Doc, and then I could like grade them. Definitely couldn't go through like every one of them, but we can go through some like key points on each one live as we're going. So I thought that would be really cool if you guys want to do that next time. I can even post the FRQ we're going to go over next time like ahead of time, so that if you want to write it early and then you just have to copy paste it like into the doc together, we can do that. So that way we can actually have time to like write out full answers, and you could spend the full approximately 23 minutes on it, whereas this time I only gave you a short amount of time just because that was kind of like a simpler question than what you would see on the full exam. Anyways, we'll do that next time. I'll post it ahead of time for you guys, so make sure to check in the discussion area before our next cram. But anyways, let's get into this. What were the answers? What did you come up with? Identify the independent variable. The independent variable would be the varying amounts of sediment in each of the tank. So actually, to get my degree, you have to do like a capstone final project experiment. And this is what I did for mine. It wasn't oysters, it was actually shrimp. But I did this when I was studying in Australia. So this was a look at Moreton Bay Island shrimp population and the effect of sedimentation on those shrimp. So this is the actual experiment that I did. So again, you would then group them into their little tanks. Your independent variable would be the different amount of sediment in each tank. Your dependent variable would be their CO2 output, measuring metabolic rate. And like in theory, what you're looking at here is that like if you put so much sediment in there that they like they're starting to die because they're unable to like breathe or commit through their do their normal life processes, their metabolism is going to go down, or they're like breathing, and then their CO2 rates would drop. So you would see like much higher CO2 rates when they were happy, and then like much low CO2 rates as they're starting to really suffer from that impacting sedimentation in their ecosystem, which I know is kind of sad to think about, but identify the control group. The tank with no particulates added would serve as the control group in this experiment, as it would tell the experimenter a baseline for a metabolic rate. Describe one variable that has not been mentioned and how it could affect the results. So on the exam, when you see a question like this, describe one variable that has not been mentioned and how it could affect the results. You have to say something and what it specifically does. So you couldn't say two things that were like not related. And this question would always be worth like 2 points, right? So describe the variable as 1 point and how it affects the results is the other. So you have to put both of those in there, and it has to make sense. So again, describe one variable that has not been mentioned and how it could affect the results. So the size distribution of the oysters, if you have all large ones in one tank and all small ones in a smaller tank, that would affect the CO2 output, right? Because larger ones in theory would produce more CO2. So you would have to make sure that the size distribution was an average by maybe weighing them, right? So you would say you'd have like the same sort of weight total in each tank of oysters, and that way you wouldn't have one that had like a ton of heavy oysters or big ones and one much smaller. The temperature of the water really matters, especially with, remember, our aquatic species are very affected by water temperature. So you wouldn't want to have like hot water in one and cold water in another. And then the particulates may actually be like kind of backfiring in this experiment because oysters are filter feeders. So are your particulates that you're using, are they like rocks and stone, or are you using like plant matter that the oysters are then eating and increasing their metabolic rate? So what are you using as your particulates, and is that actually backfiring and changing your experiment? And then describe a modification that could be made and how it would affect the results. So you could monitor the temperatures. You could purposely change the size of them. You could increase the amount of particulates or a pollutant found. You could find the lethal dose 50 of particulates. So again, the amount of particulates that would cause 50% of the population to die. So that's a modification that you could make to this experiment, and how that would affect your overall results. And again, if you are coming up with any questions as we are rolling through this, definitely put them in the chat. I do see some questions. Let me check those out here too. All right, so let's talk about our dependent variable, and could you just write the oyster's metabolic rate? You would want to say like oyster's metabolic rate as measured in CO2 output. Yep, and Miriam's definitely saying you have to be very specific with your dependent and independent variables because your graph has to match. Yes, that is 100% it. Your dependent is your Y and your independent is your X. And that is what I really wanted you guys to get out of this little, Miriam is on fire. She's got it. That is exactly what I wanted you guys to get out of doing this. Anytime you make a graph, you're going to want to put your independent variable on your x-axis. So you could have said, like I put tank 1, tank 2, tank 3, because we know from that other chart that like tank 1 had 0. We can go back and read the exact amounts, but like tank 1 had 0, tank 2 had 25. But if you wanted to on your x-axis, you don't have to identify the tank numbers. You could have just said 0, 25, 50, 75, 100 parts per million on the bottom. So you could either say the tank number or the particulate number on the bottom. And I purposely kind of wanted to include 3 row or 3 columns of numbers here to throw you off, right? Because you saw those 3 columns and you may have been like, what goes on the X and what goes on the Y. So I kind of wanted to throw you off on purpose on that one. So again, so those could say tanks on the bottom, or it could say 0, 25, 50 and measure it in that way. They're the same numbers. Your CO2 levels, your dependent variable, what we're measuring, that's going to be on the y-axis. So you're going to put those up. Remember, you always want to use even measurements. So on either one, you're never going to have like it go 25, 50, 100, 200, 400. Like it's always going to be even measurements as you're moving up, or even increments, I should say. Oh, someone asked, does it have to be a bar graph? And the answer is pretty much, yeah. We are not measuring change over time. So when you're going to measure change over time, that's when you would do a line graph. But these bars are not connected to each other in any way. They are independent statements of fact, right? So there are independent things that happened from each other that you're measuring on the same plane or looking at the same, degree of CO2, but they're not dependent. So you can't connect them as in like a line graph. You also are not looking at parts of a whole, so you wouldn't be able to use a pie chart. Really, a bar graph is going to be what makes the most sense here. Can we just plot the points on a graph? So You could just put the dots up there, not connected. But visually speaking, that would be sort of confusing to look at. For the most part, a bar graph here is really going to be like, I know sometimes we would do like a scatter plot where you just like put the dots independently and you look for a correlation. So you could, it wouldn't be like really wrong, but it would be, I would say, more correct in this term to use a bar graph for sure. And then again, anytime you want to make sure you label all of your axes. So you could write it in the y-axis, you could say 100 parts per million, 200 parts per million, or you could just make sure it's labeled on the outside, and that kind of encompasses everything then. So CO2 levels in parts per million, and that applies to everything that it's over in that area. And then your title needs to be an independent statement that includes your X and Y components as well. So you want to have your independent and dependent variables mentioned in your title. So sedimentation levels on the metabolic rate of oysters as measured by CO2 in parts per million. Your title is not going to be something funny. It's not going to be something clever as much as we would want it to be. It needs to just be really dry. It has to include your independent and dependent variable. And it has to include your like units as well. So like your parts per million there. And again, you might not be asked to graph. And it's kind of hard for us to tell because we haven't done this type of question very much at all yet on the AP exam. That's why I felt like it's better for us to just kind of shoot high. Like this would be very high level if they asked you to like create this whole thing for them. But as long as you feel comfortable doing this, then whatever they do ask you to do, whether it's analyzing a chart like this, or creating something similar to this, you're going to be comfortable with it. So I would rather like aim high in your understanding of scientific experimentation, and then on the test they ask you something easier. But yeah, so that's kind of what we're seeing. So it is pretty much time to go, but I'm going to leave you with one more thing. We don't have to do this right now. You can come back to it. This is like the other aspect that you could see on the exam here is they might pose a problem and want you to create an experiment. So if you are creating an experiment, I want you to stop and think about what we talked about. Is it a real experiment? Do you have an independent variable? Do you have a dependent variable? Is it clear? And the more clear you are on the exam, the better. And again, this isn't an essay. We're not looking for you to write out something beautiful. You could be very clear with your response, like just say independent variable, dependent variable, and then write it out in a sentence as well. You know, these are people, they're humans who are grading your responses. So if you make it easy for them to find, they're just looking to check off tick marks, right? So if they're looking for you to state an independent variable, write it out, independent variable. So if they do want you to design your own experiment, really make sure that you're breaking it down in your head, that you have all these pieces, and then, you know, where are they? Is it easy for the reader to find what you're trying to say? And is it easy for them to see that you know what you're talking about, right? So that's kind of for this exam in particular, you want to make it really easy for them to tell that you know what your independent variable is. So again, here's another question for you to practice with. Scientists are worried about increasing acid deposition, causing harm to forests across the East Coast. Design an experiment that would help them assess the potential harm, and how would the data be collected, and what information would be present. So if you're looking at a bunch of different forests on the East Coast, how do we know that they're being harmed by acid rain? What experiment could we design to test that? How would we collect the data, and what information would we be collecting? And I want you guys, our time is up here, but Miriam is going to be in Discord for the next 30 minutes. I would love if you guys could post your answers to this there and have a discussion as far as like what would be a good experiment. What could you do? And again, be creative, pull in different things that you've done in school or class. What experiment could you do? And this one's a little bit challenging too, because it's not in a lab, right? What could you do out in a forest to assess the potential harm? Is it quantitative? Is it qualitative? And again, what information would you get from that? Okay, guys, thank you so much for joining me. I hope that this was helpful and thought-provoking, and I will see you guys again soon. And for the next ones, we definitely will look at specific College Board kind of layout for those, but this one was a little bit different. Hopefully it was fun. Hope you have a good rest of your weekend, guys. Bye.